Synergistic effects of apitoxin and honey on gram-positive and gram-negative bacteria by Jack Greenwell.
And Jeremy Hu. Our adult sponsor, Miss Young, helped us by ordering all the materials we needed for experimentation.
This study aims to test synergistic effects of bee venom and honey against Salmonella and Streptococcus bacteria.
This antibiotic could be used in the real world as a topical treatment when applied to the skin for infections.
This is especially important now as antibiotic resistance is becoming an increasingly preponderant trait among bacteria.
Question. Can the effectiveness of aflatoxin against Streptococcus mutans and Salmonella enterica bacteria be improved by pairing it with honey?
We believe that if apitoxin and honey have significant synergistic effects, then combining them will result in greater inhibition against Streptococcus and Salmonella.
Variables and control. Our independent variable is the solutions that are being tested. Our dependent variable are the measured diameter of zone of inhibition.
Our controls will be that we will be using the same solutions, the same two sources of bacteria, the same type of inhibition disks, and the same incubator and incubation conditions.
Materials. We'll be using 2 strains of bacteria, inhibition discs, our 3 solutions, caliper, and agar plates.
We will also be using pipettes, petri dishes, laboratory safety equipment, bleach solution, and ethyl alcohol.
Procedures. For preparation, we'll disinfect all testing area and equipment. We'll also culture our bacteria and create our solutions for testing.
We will then begin testing by placing our inhibition disks in each of the solutions to soak up the solutions, and then placing disks into each strain of bacterial cultures. We will place each of these 3 trials of bacterial cultures into the incubator for 5 days. Data collection will be performed on the final 3 days.
Safety, risk, and disposal. We wore laboratory coats, masks, gloves, and glasses. All experimentation was also conducted in a biological hood. All biohazards was disinfected with bleach and thrown away in biohazards waste bins.
We also washed our hands before and after contact with any of the materials used in the experiment. This table shows the overall averages of our results for each of the tested solutions.
These are results displayed as graphs.
We performed both an ANOVA and t-test for each trial set to ensure statistical significance of the data, and our data was shown to be statistically significant.
Discussion. Honey proved to be very effective against Salmonella. It was unexpected for the bee venom to not work as well as we expected, as it went against our background research.
These results may have occurred if the bee venom was denatured before shipping, but this is pure speculation. Synergistic effects between the 2 solutions tested were not present or severely lacking.
Conclusion. The data disproves the hypothesis that apitoxin in honey has synergistic effects.
In the future, we would aim to mitigate our weakness of poorly swabbing the Streptococcus bacteria. On the other hand, we would aim to continue on with our strength of using a digital caliper to allow for precise and accurate measurements of inhibition zones.
Furthermore, we'd also continue to testing in other laboratory models or even other venoms, such as wasp venom.
Thank you.